The outline is indicated here. Plant-based polyphenols, phytochemicals, phytoingredients have gained an important significance owing to several associated health benefits. A predicted boom in the polyphenol market owing to the increasing demands and market size support continues. Consumer trend also support this, so we expect that vitamin E, polyphenols, flavonoid market, amongst others, will continue to expand. According to data from Frost and Sullivan and other marketing research. With regards to polyphenols are classified into different subgroups here as indicated here. Stilbenes and lignans are non-flavonoid compounds. Flavonoids includes flavonols, amongst others, and isoflavones. Quercetin and camphorol are two very important and most studied flavonols. Regarding phenolic acid and derivatives, caffeic acid, synapic acid are widely investigated hydroxycinnamic acids. Of course, there are other hydroxybenzoic and other hydroxycinnamic acids, but in the context of this talk, I will talk about synapic acid and derivatives. Given the widely accepted antioxidant capacity of the phenolic acids and various other polyphenols, including the camphorol derivatives, our project goal is to enhance the conventional processing indicated here. Conventional pressing generates the pressed oil and also the crude oil, further refined to produce a refined oil. Biodistillate is a byproduct here in this process. It also contains significant cannelol. So back to the project goal. Our project will focus on pretreatment. Example of thermal pretreatment is the application of heat, oven roasting, or toasting treatments. We aim to apply integrated approach for antioxidant enhancement in the oilseed industry, and our past presentations have focused on extraction of canola press cake and mustard press cake. Important oilseed crops includes canola, rapeseed, and mustard seed. The production data for mustard, so Canadian data for mustard production is indicated here. Canada is one of the top 10 producers of mustard. Oriental, Brassica zuntia L, yellow, Sinapis alba mustard are among the important varieties that are currently produced in Canada. Widely used as a condiment, spice, emulsifying agent in meat, sausage, dressings, meat, etc. Standard grade according to specifications, grade 1, 2, 3, 4, and of course lower grade is referred here as sample grade, which we are using for the research purpose. Commercially, the products indicated here are available. Cracked seeds, flour, cracked deheated flour, bran, mustard oil. Mustard oil is very widely used in some of the countries like India and Nepal. Synapic acid and its derivatives are present in high concentration in oilseed crops belonging to the Brassicaceae family. Roughly 73% of free phenolics and 80% of total phenolics in mustard is synapic acid. In white seeds and black seeds, of course, there are some imminent differences. 3,4-dihydroxybenzoic acid, ferulic acid is reported in white mustard seed, whereas the presence of rutin is also reported in black mustard seeds. Other compounds, vanillic, syringic, picoumeric acid is also found in mustard seed. Camphorol derivatives camphorol glucosides, camphorol acyl glycosides are also reported to be present in mustard. A total of 91 flavonoids and hydroxycinnamic acid derivatives were detected in Brassica napus L in another study. So this is interesting, and with the number 39 camphorol derivatives, there is a wide scope for investigation. The structure of the important synapic acid derivatives that I will be following as part of this talk and following in research is indicated here in the slide. Synapic acid, the choline ester of synapic acid, synapol glucose, and canolol or 4-vinyl syringol is indicated here. 4-vinyl syringol is also referred to as canolol and is generated through decarboxylation of synapic acid. The occurrence of free synapic acid is indicated here in this slide, and it is important to note that the free synapic acid is significantly lower as compared to synapine. And our past work, we have focused on synapic acid. Our hypothesis was centered around the gap between the low content of synapic acid and the high degradation of synapine. We speculated that synapic acid derivatives contributed to the formation of canolol. So why study thermal pretreatment? Thermal treatment induces structural changes, further inducing chemical and functional changes. Thermally treated mustard seed might have pronounced antioxidants. Shelf life, storage stability impacts formulations and use, and also impacts the flavor. An example is in one of the study, it has been reported that mustard gives a typical flavor due to the roasting treatment. The content of canolol in canola oil before and after roasting of seeds is indicated here in the slide. As you may see here, after roasting, the canolol content is increased, proving the efficacy of roasting. Other pretreatments have supported the fact that canolol and synapic acid derivatives have been impacted. For instance, pretreatment, superheated steam, microwave have been used to impact recovery of canolol and synapic acid derivatives. An example is other oven treatment or thermal treatment. The highest level of trans synapic acid was found in the oil, roasted at 160 for 15 minutes. The other study used 180 for 20 minutes and reported the highest sample canolol in both rapeseed and mustard seeds as well. Our laboratory investigated canola seeds of different varieties, expeller, press, cake, laboratory derived cake as well. And about 180 to 200 degree with the temperature time combination 10 to 15 minutes was successfully used to produce the maximum canolol. Another study used 160 degrees C to enhance the canolol content. Pre-treatment such as microwave, oven, steam has been successfully used. So in this study, average phenolic acid in pre-processed canola seeds with different pre-treatments were compared. As you see, compared to the unprocessed, roasting was successfully enhancing the content of the synapic acid and the canolol content, and of course the total phenolic in this study as well. So these are some examples of the successful integration of roasting treatment. For the successful, meaningful integration of pre-treatment, several factors must be considered. For example, structure-function roles, stability, extraction parameters, isolation, and of course, whether we are using the seed or cake or other byproduct must be considered. So with this, I would like to introduce the project objectives. The project aims to study the effect of different extraction conditions using air frying oven and adapted rapid oxy oven on the recovery of the marker compounds. We also aim to determine the cannellol and degradation products associated with the synaptates in the mustard samples. The significance of the study is outlined here. The antioxidant activity of main phenolic compounds of pre-treated mustard and degradation products due to roasting is largely unknown. Mustard recipes usually includes thermal processes such as cooking, baking, roasting, pot frying, pan frying prior to the addition of oil, water, and other ingredients. And hence, identifying the degradation products of thermal pretreatment is applicable to food processing and food consumption patterns, and thermal pretreatment is an alternative approach. So with this, I would like to introduce you to the paper that we are here as part of the, this work was conducted as part of the project on pre-treatment, preheating of yellow mustard seed. The title of the talk is indicated here. The methodology, we have used air fried and rapid oxy. Temperature 160, 170, and 180 was used under different experimental conditions, time combination ranging from 5 to 20 minutes. And degradation was monitored by means of HPLC DAD and LCMS. We have used yellow and oriental mustard products, commercial products. Now, you may be aware of Rapid Oxy 100. Most of our colleagues are using this to assess oxidative stability. We have adapted this in this particular study to air frying preheat treatment of mustard fractions. The experimental design is indicated here. The low-grade mustard seeds are air fried and afterwards using the oxidative apparatus, it is the oil to separate to the oil and the cake, and investigated for phenolic extracts using the HPLC and LCMS. LCMS was conducted in positive mode of analysis, and the formula generation, the scan mode, was set to the MS mode. Fragmentation patterns were compared with the literature values, and we are still continuing some more analysis, some more work in this regard. So the objectives of the study particular paper is here, indicated here, to examine the effect of the conditions in oil. So oil-soluble phenolics were investigated, and then to assess the effectiveness of air fryer as an alternative preheat treatment for the formation of cannellol and other lipophilic synaptates. The other objective is to assess the effectiveness of air fryer as an alternative preheat treatment for the formation of other synaptic derivatives in the residual cake, the oil substrate cake. This diagram indicates the result. In the cake, it was proven that synapine was degrading, whereas in the oil, at 160, as you see, an increase of cannellol was apparent. There was another compound which was detected at 17.1, indicated here, which was again increasing. A third compound was also detected at 15.7 retention time, and this compound also increases. For the 180 degree C study, cannellol also increased as compared to the 5 minutes. The other two compounds detected at 17.1, 15.7 also increased. So we are investigating these unknown compounds. The UV DAD spectra is indicated of the unknown compounds are indicated here. This is the UV spectra of canolol. Canolol is predicted to react about 3.6 times faster in lipid media than in the aqueous solution. However, there's still a lot of information about the chemistry of canolol that needs to be investigated. Degradation aspects, stability aspects, dimerization conditions, and molecular modeling. In one of the study, canolol dimers was found to be more stable compared to canolol. The study compared the stability of canolol and its dimer for a period of over 5 months. Another study indicated that synapic acid, when exposed to air, is converted to thomasidiacic acid and other compounds. So we still need to learn a lot about chemistry of canolol. The LCMS analysis result is indicated here in this chromatogram. We were able to detect synapine, syringic acid, synapic acid, the dihydro synapic acid, thomasidiac acid, canolol, and synapic acid dimer. Other camphorol derivatives, 3 camphorol derivatives as indicated here, were also detected using our LCMS analysis, and we are continuing our work. This work is still in progress, although it was interrupted by the COVID situation. So, impact of thermal treatment. The proposed scheme of the degradation of the synapic acid derivatives using thermal treatment at 160 to 180 and corresponding formation of selected breakdown products are indicated here. Synapine, the choline ester, and synapol esters and other glycosides undergoes thermohydrolysis. Synapic acid is the product. Synapic acid also generates dimers according to the temperature, pressure, and other conditions. Examples include syringaldehyde, dihydrodimers, thomasidiac acid. Synapic acid is decarboxylated to form canolol, and canolol generates further other dimers, produces other dimers as well as other products. An example of a product is a phenolic adduct. For an example of a proanthocyanin adduct is indicated here as an example. These adducts are known to be fairly stable. Other products may be Maillard reaction and phenolic adduct as well. So it is interesting to investigate what are the other products that is associated with canolol as well. So the result of the current study is indicated here. Canolol was readily extracted into the lipid, confirming its lipophilic nature. A hot air frying treatment enhanced the lipophilic phenolics in the oil. Maximum canolol was obtained at air frying temperatures of 180 and 160 with a time combination of 10 to 20 minutes. Furthermore, 170 degrees C and 10 minutes was also suitable for the maximum recovery of canolol, and we are still working on this setup combination for 170 C. Other unidentified phenolic compounds which are impacted by the pretreatment and increase with the increasing time and temperature combination until up to 180. So we are aiming to identify these unidentified compounds. No canolol was present in the pretreated sample. In some other samples, we have seen some traces and some presence of the presence of cannalol, but not in this one. The presence of other cynapoyl and camphorol compounds is confirmed from this product, that mustard product that we have used. It's interesting because camphorol compound, one of the camphorol sapouroside, is recently identified as a contributing factor for the bitterness of canola rapeseed proteins. So in summary, pretreatment is a simplified technology, dry, aqueous, solvent-free. Pretreatment prior to cold pressing or conventional oil extraction enhances lipid-soluble phenolic compounds. Otherwise, just pressing the oil, most of the phenolic compounds remain in the cake and only a small proportion is transferred to the crude oil. Pretreatment can be used for other oil seeds as well. For future perspective, limited data on flavor synaptic derivatives is available. So we are very interested to study about the flavor aspect of the camphorol and synaptic derivatives and canola derivatives as well. Monitoring these chemical and structural changes during heating, again, is interesting to study. Many properties remain to be examined, heat transition, kinetics, and molecular modeling as well. So with that, I would like to thank you for this opportunity. The funding agencies are acknowledged here: CAP, the Saskatchewan ADF project, and other collaborators, including Professor Ghosh, University of Saskatchewan, NSERC Discovery Grant, Gates Scholarship. I would like to acknowledge my collaborator and mentor, Professor Eskin. I would like to acknowledge Olamide, my PhD student, Afra Ruchira, former technician Jingbang, and former postdoc Dr. Achari. And I'm happy to take your questions now. Thank you for this opportunity.